Thanks for having me. I actually really like Little Rock. I like the vibe. And I hope what I talk about will be useful. And I've really confined it. I've sort of separated the proteomics part from the biology. Also, I'm thinking I might be skewered because you're going to look at some of the things that I've done, which are kind of ad hoc and intuitive, and say, what were you thinking? So we'll see. We'll see if that happens. It would be good to know if that happens now, it's better than if the reviewers do it. Okay. So this is basic stuff. We all know that, a lot of us in the world of biology and molecular biology are interested in identifying protein-protein interactions. So we have our sort of set of low throughput methods that we use, co-IP, Westerns, yeast 2 hybrid, various physical methods. And then there's a lot of microscopy methods that I run across, quite finicky and a lot of work to set up. But if you want to see two molecules interacting in a cell, particularly in a live cell, these can be quite useful. And then there's the high throughput ones. So this one's sort of been around for quite a while, and we've done some of this, and with some success, in fact, not a ton, but just like expressing things in HeLa cells, co-IP, MS of some sort. I've done a lot of yeast 2 hybrid in my life. I've never done phage display. And so what I'm talking about today is proximity labeling. That's the kind of general term for it. So here's the basic idea of proximity labeling. And in this case, the term is used interchangeably sometimes with BioID. BioID really refers to a subset of proximity labeling experiments, and this falls under the BioID umbrella. But the basic idea is, of course, you fuse your protein of interest, and we can call that a bait, to a protein ligase enzyme. In this case, it's a particular ligase if it's BioID. And so we're fusing it to our bait to something called TurboID. So this is a biotin ligase, or maybe more accurately, a biotin activating enzyme. It converts biotin, which is just, vitamin, what is it, B7? It converts it into an unstable radical, which then will react with lysine residues on nearby proteins to form a covalent bond. So it's not a specific targeted event like phosphorylation where it recognizes the binding site. It's literally just leaving a trail of these unstable biotin molecules that then attach to whatever they're going to attach to. And that radius, it's estimated, it depends, but you'll see some reports that it's 10 nanometers, so only maybe a couple diameters of a protein, to more, I think, accurately, which is here 35 nanometers. So that's its sphere. And the closer this other protein is, the more likely it is to be biotinylated. So as I just said, the ligase covalently tags neighbors. And biotin is one of the things you can tag with. There are other versions that use other modifiers or other ways to generate the active biotin. Then it's just affinity purification of the tag, and you identify the enriched proteins. So this just shows the schematic. So biotin is great because it binds to streptavidin. You can buy streptavidin beads. And that binding interaction is like one of the lowest KDs ever measured. It's super glue. In fact, you have to go to great measures to get it off the biotin bead. And what we've done with Arkansas is we actually don't try to separate the biotin. We don't try to compete it or boil it or put it in some, extreme solvent. So you have, this is what's going on in the cell in steady state, because what I'll mention is that we tagged our bait using CRISPR. So this is an endogenously tagged functional protein. And it's at physiological levels, which has good points and also bad points if your protein isn't very well expressed. So the binding reaction takes place. So the beads bind to the red biotin. Those get pulled down. The stuff that wasn't biotinylated, you wash out, at least hopefully. And you end up with these beads. And then this is actually what we send to Arkansas. And then they carry out trypsinization and a bunch of LCMS steps. And the one thing that this is a little bit accurate about is that this biotin right here, the biotin and the portion that's directly attached to the biotin that isn't cleaved off, that probably is just staying on the beads, because it's my understanding that you do not detect very many biotinylated peptides here. And that was a total misconception I had when I started this. I thought, oh, we'll get biotin tagged peptides. No, you get enriched peptides because they came with your pull down. And then what I'll talk actually almost the most about really is this next thing here, which is really my efforts to stare at the data for thousands or, probably close to thousands of hours, trying to figure out what it meant and how to filter it out and what's good and what's garbage and that sort of thing. Okay, so the main advantages of proximity labeling. So someone asks you, so I've tried to give this talk from the standpoint, I guess, of a client. So a lot of these are titled by a question like you might get asked. Should I try it? So, okay, the main advantages are detection of weak and transient interactors. That's huge, actually. And I'm in the worm system, and in case you don't know, worms have this cuticle. It's a pain in the neck to do biochemistry in worms. A lot of us have run screaming when we've tried to do it. We're all afraid of biochemistry. And so breaking the cuticle, right, and then doing an IP pull down, that's asking a lot. Just solubilizing the worm and getting the lysates, you're going to disrupt a lot of interactions. So it's not like nice, soft, squishy cells that, you just sort of do something with a weak detergent and those, it's at least some of those direct physical interactions survive. So, it's great for weak stuff because it's all happening in vivo. You don't have to preserve that interaction. It's really impressive in my experience to detect signal within a lot of potential noise. So the proteins I'll tell you about that we've looked at, they're only expressed in one tissue of the whole worm. We can't dissect it out. The worms are 1 millimeter long, right? So, and they're not expressed at very high levels. So it's a pretty low proportion of the total lysate. It's like, you took your cells of interest and diluted them with a bunch of other garbage, right? And you still get the signal out. So that's what we're asking the system to do for us. Disadvantages, there is some work at the front end to build strains, test conditions. There's always work at the front end of any assay, but that's one of the things. Dealing with false negatives and positives. Actually, false negatives aren't a problem because you don't see them, right? I mean, if you knew they were supposed to be there, you might be concerned, but I don't worry about false negatives. I worry about more like, I don't want to waste time on the long list of proteins potentially I might be getting because everything I do afterwards is some sort of functional assay. It could be genetics or cell biology or more biochemistry, but that all takes time. And you do have to deal with potentially semi-long lists. It depends how you define long. I'll show you what sort of things I've been getting. Okay, so then the next question you might get, and this really is not a question you should get. I mean, the people who are asking you to do the experiment should read a few review articles and figure this out themselves. But, what type of proximity labeling should I do? And I'm not going to go through the various versions of it. There's options where you use different types of enzymes or substrates, and it should be guided by the experimental system and the biological question that you happen to work on. So we used an enzyme called TurboID, meaning we tagged, I guess we made about 8 or 9 separate CRISPR strains, where we tagged TurboID onto the tail end or the end terminus of our proteins of interest. So this is a biotin ligase derived, or you could say evolved or engineered from BRA. It's a bacterial protein. It's a biotin ligase that targets in a very specific way, sort of like a kinase, okay? But it's been devolved so that it has much higher catalytic activity than the original BRA, or even the original BioID that came out about 10 years ago. It works at a range of temperatures. It is very non-specific in its target activity, sort of like I was describing earlier. That's what you want. You want the specificity to come from the bait and what binds to the bait or is in the vicinity of the bait. You don't want it coming from the ligase itself. You want that to be completely indiscriminate. It's relatively small. There's a smaller version called mini turbo that we decided not to go with. It makes use of endogenous biotin. So yeah, vitamin B7. So you don't have to add anything. You don't have to get anything in past the membrane, which is an issue with some of the substrates for these types of approaches. It's used genetically encoded, so physiological levels. it's really going to be best for steady state. Okay, so what we did is we took worms, we took a mixed stage population, and there's advantages and disadvantages of doing something like that. But this isn't like, if you wanted to do a really short time window or a time course, that might be hard to do with Turbo, and you'd probably want to use another approach, like probably the second most highly used approach, or maybe it's more, is something called Apex. It requires hydrogen peroxide to activate it. And so, it's not active until the second you want it to be active. And then you just let it do its thing for a very short period of time. Here, we're letting Turbo do things for hours, days even, potentially. Okay. So our focus were these 5 proteins. And like I said, I'm not going to really talk at all about the biology, but 2 of them are protein kinases. They all have really good human orthologs. NECL3 is a kinase. NECL2 is another kinase. NECL2 probably phosphorylates NECL3. And they both have these binding partners, which are named MOLT2, 3, and 4. They're anchor and repeat proteins. They form scaffolds. It's what localizes these kinases correctly. And they roughly form 2 complexes that we knew about from cell biology, but they also seem to have some interactions as well as some overlap in terms of their localization. So you can so this is AlphaFold, and I'm not going to talk about AlphaFold, but what I will tell you maybe at the end is that I think the best post hoc analysis and pipeline even that you might provide as a facility, and I know you don't want to do this, is a thorough, rigorous AlphaFold AlphaFold study of what you've pulled out and whether those might directly interact with the protein that you started with, because or do they interact with each other? That gets to be a lot of interactions to test, but that's what supercomputers are for. And it's very valuable. Okay, so they can form something that looks like what I've shown here. All right. So the experiment was we would take the NECLs, these kinases, the MOLTs, these anchor and repeat partners again, and fuse them to Turbo, either on the C terminus or the N terminus. I will mention that the C terminal fusions work so much better than the N terminal fusions. I have no idea why. They're expressed at the same levels, but if it doesn't work on one end of the protein, try it on another, and maybe even in the middle. I don't know. But that was our experience. So here's the idea. You really expect cis biotinylation or self-biotinylation, right? This is NECL3 with Turbo. It's going to hit itself because it's going to create these unstable bindings, and they're going to immediately attach to lysines on NECL3 itself. NECL3 binds to MOLT3, so you also expect this trans biotinylation event to occur, another great internal control for your experiment. And since these sort of form a complex and do overlap, you might expect these other guys to be biotinylated, although maybe to a lesser extent. So this is what you really want out of it, right? You want to find things that bind NECL3 or are somehow associated with it, even indirectly, and pull those out of your screens. Okay, so our experiment was we CRISPR'd the TurboIDs, our control, and I'll talk a little bit more about controls because this is a really tough issue. We use just the wild type strain that's called N2 for C. elegans. So it's really a no turbo control. That was our main control. We make lysates from mixed stage worms, in part to cast a wide net, in part because we're lazy, and that was sort of the easiest way to do it. We found that a 1 hour incubation time of lysate with the streptavidin beads was optimal. More than that, you start to get a lot of sticky junk. We send the beads directly to Arkansas, and that spend a lot of time. So this is actually one of our better gels for what we get when we use streptavidin is essentially like an antibody for a Western blot. And what we're looking at is wild type no turbo, NECL2 turbo, and NECL3 turbo. So one thing you'll notice is right away there's this big band here, and maybe it's a little hard, but there's some other bands in there too. Those are endogenously biotinylated proteins in C. elegans. And every organism has a subset, usually a very small subset of proteins that have biotin attached to them. In C. elegans, and I think in some other species, they tend to be these carboxylases. I have no idea what their functions are. I know they're quite abundant proteins in general. And it really just comes down to 4 proteins, some of which co-migrate, that are biotinylated. They have multiple isoforms. So on some of our gels, we often just see one predominant band. On others, we see more distinct bands. So what you would hope to see, because you've now added a new biotin ligase to this gamish, to the cell, you'd expect to see, well, there should be, in addition to this one, there should be other bands. And we do see some other bands here. And so that's a little bit promising, right? And so this was, and then, you put a tag onto NECL2 and NECL3 so you can see that they're there. You do actin to see if you've got equal loading, et cetera. In fact, these carboxylases work just as well as actin to know if you've loaded it. This is just the experiment where we incubated for different amounts of time, either the control or the NECL3 turbo. And then we just sampled the lysate, ran it on a Western to see if any of the biotinylated proteins were left, or had they all been sucked up onto the beads. And the answer is pretty much right within an hour, it's just about done. And longer than that doesn't really help. You can even see down here, within an hour, we've got quite a bit less of NECL3 because the biotinylated NECL3 got sucked onto the beads too. It's not all biotinylated, you can see, but the stuff that is biotinylated, that also gets pulled onto the beads. This is more like what might represent some of our typical experiments. I think this was experiment number 6. So this is NECL2. This is a control turbo that I'll tell you about that we used. And this is NECL2, 3, 4, et cetera. One thing you're going to notice right away, and believe me, we noticed this, is that we looked at N2, and then we looked at these and we said they look the same. This experiment did not work. Okay, yeah, we saw something here, but on most of our gels, for whatever reason, we didn't see much. And I really was worried that all the mass spec would do was read these things and there would be nothing else to see. I thought 99% of the reads are going to be these 4 carboxylases. That wasn't true. It turned out we got great stuff from these types of experiments. And so what I'll come back to is, when people say, God, I want to know if should I even bother to send you my sample? The answer is, it doesn't cost that much, and you literally can't tell from the gel unless your gel looked like you really screwed up and you don't have anything there. What do you have to lose? And as I said, we had great stuff from this, and that's just sort of more stuff. You can see these are pretty low abundance proteins, sometimes very hard to see even. Okay, so how many technical replicates do I need? So, okay, that's an interesting. So here's what I'll say about that. Okay, it's a fair guess. My answer is 3, and I'll show you why I think that. So we started with 3 just to see if the experiment would work. So what I've done here is I've taken all of the proteins that were in NECL3 experiment 1, are 3 technical replicates. I take the intensities of every protein where it was in both of them, right? And I'll talk about that too. And I just plot them onto an XY and see how well they correlate. So there's a few thousand dots here, okay? And you can see rep 1 compared to rep 2 looks pretty good. Rep 1 compared to rep 3 and rep 2 compared to rep 3. So it looks like the 3 things are doing pretty similar things. And when you look at a spreadsheet, yeah, the numbers really do track. This is just proteins confined that were up 2-fold relative to our no turbo or N2 control. And those also look good. So things, to me, the replicates are really pretty replicated, right? So then the other question is, okay, well, they track in terms of intensity, but how well do they overlap in general? I mean, that's just the ones that overlap. What about overall overlap, like they detect the same proteins? So I'll show you a few different results. Most of them look sort of like the good ones here. So this is N2 experiment 1. So this is one of these overlays. It's sort of a Venn diagram with 3 things, but you can only see one thing, right? So this is sort of those proportional Venn diagrams that sometimes are helpful. I kind of like it for this experiment. So this means it was present in 1, 2, and 3. That's 91% or 3,300 out of 3,685 proteins. So they're finding the same things, right? And the requirement here is it had to show up in all 3 replicates to count it. And you can see very little of these other possibilities, right? So that looked good for experiment 1. Experiment 6 looked pretty good. The one experiment where it didn't work was experiment 7. It was specifically for NECL 3. I mean, the N2 isn't as good as these, but it's not bad. This one clearly, like, there was something wrong with big time with one sample, and that other sample wasn't great either. And here's the gel, right? This is replicate 2. That looks okay. Well, there's obviously something we screwed up there. Why did we send you the samples? I have no idea, right? We shouldn't have, right? So that was a little bit of a waste. So that was on us. Okay. So generally, I think 3 replicates is sufficient, but pay attention to red flags. If things don't look good, it's, you know, it's not just you forgot to load the gel right. You really might have a bad sample. So how do I know my samples are good? Your gels look consistent across technical replicates, but like I said, in reality, you don't. So this is this is something we published from here. And so this is a protein. This is another protein that's connected genetically to some of the proteins I've told you about called PTPN22. Here's the N2 control. Now you can really see this is a beautiful gel. That grad student, she left the lab and no one can run Westerns like her. I don't know why not, but like she's the only one that could ever get it to look like that. This is PTPN22 turbo. Right away you can see, wow, now I really convincingly see all these other bands there. It's really doing something. So that's the input. This is the lysate. So clearly when you see that, it's more encouraging than when you see this, right? Like this really doesn't look good. This was our best experiment, literally. This was our golden experiment, it turns out, and that's what the gel looked like. And that's why I say you can't know until you run it. What controls do I need? So this to me is a pretty tough question. And the answer is you may not know until you do some experiments. That's the way it goes sometimes. Obviously, for us, like a no turbo N2 or wild type was a good starting place. And it's not even a bad finishing place, but you'll see we threw something else in too. You know, so that also might just be your starting cell line that you haven't transfected or whatever else. That's a totally, I mean, minimally, you're going to have to do that to get a fold change. Alternatively, you might want to express some version of the BioID like turbo in the same cells of that organism, but where you don't have your bait attached to it. So essentially a no bait control, or, you know, we just have a strain with GFP tag to turbo. So, you know, GFP just kind of floats around. At least that's the idea. So the reason you need this is because obviously this is what we want. Unfortunately, this is what you will also get. Guaranteed 100% of the time you will get this. So here's your turbo. It's going to hit non-specific proteins. There are proteins that are relatively highly abundant. Maybe they have, you know, plenty of lysine surface residues, but for whatever reason, they love to be biotinylated. It doesn't matter what bait you stick on this end, a lot of the same proteins will get biotinylated. Now that may vary whether you're talking about cytoplasm or nucleus or this cell type versus that cell type, but these are common garbageome type hits that you get from TurboID. And I imagine these become more and more known, and you know, you can maybe exclude them without doing too many experiments. So what you need is a non-bait specific control. So like I said, GFP turbo, in theory, it should be hitting a lot of the same ones. And we got this strain from another lab. What they did is they put this promoter on it that was epidermal specific. Our proteins are also epidermal specific. One downside is this is expressed at much higher levels than our proteins, and that can be an issue. So, okay. So a question you do need to ask about these is, and it doesn't mean that any of these things mean don't do it. You're still better off doing it, but it's things to consider. Is this control turbo and your bait turbo, are they expressed at the same level? Are they expressed in the same tissue? That is pretty important. Are they localized to the same subcellular compartment? If your protein's in the cytoplasm, or in the nucleus, and this GFP is mainly in the cytoplasm, it may not be a great control. There's different proteins in those compartments. Of course, you can start to go crazy, right? You can say, well, I need one that's, you know, in the Golgi. Well, then it might be hitting some of the proteins you're hitting, but yours are real, right? And so you kind of have to balance it, you know? And the other point is it in the same genetic background? So our wild type is not exactly the same as the wild type we got from this other guy, right? And you know, it's like HeLa cells in your lab aren't the same as HeLa cells in someone else's lab, at least not exactly. So that's going to cause some differences too. So it's best if you can keep it isogenic. And I wouldn't say we had the ideal control, but it wasn't bad. And we tried to make some others, and for technical reasons, we failed. And that was that. So we went with what we had. How do I know if my experiment worked? This is a big one. Okay, first point, did you get your bait protein back? That should be your number one, all right? Number two, did you get known bait interactors back? One thing though is a lot of people might do this experiment because they don't know what it interacts with. And so they might only have the bait to go with. But it's nice if you know some other things, as we did from genetics and biochemistry and cell biology. Okay, so this is sort of an example of fold changes. So this is, again, I'll remind you, this is the complex, so 5 proteins. This is sort of the idea of cis biotinylation versus trans, okay? And so these are from 2 different experiments. I just sort of combined them. And so this is what was detected up here, color-coded, NECL2, 3, MUL2, 3, and 4. And these were the turbos, right? And so the diagonal is the self, and that's also in mold. So if we look at NECL2, okay, 228-fold upregulated. Now this one has the Berry bonds asterisk, and I'll explain why. It's because I had to make a guess about the fold change, and that's because it wasn't detected in the no turbo control. And I'll talk about that briefly. Not so much how I did it, but again, that you're going to get a lot of these things. But what you can see is, okay, it also detected NECL3, MUL2, MUL3, and again, this was an all or nothing, so that MUL4 also has the asterisk, at least for some of them. And if you go down here, you know, these are pretty good fold changes, right? I mean, this is nearly 200. It pulled out this partner 65. This is over 100. It pulled out its partner nearly 20. That's great. And then in most cases, they would pull out each other. And we already knew, and it sort of made sense too, because the fold change correlated whether they were in this complex together or this complex together, at least to some extent. All right, so this is looking more like a, you know, I sort of took what I get directly off your website, which is where I steal the volcano plots from, and I'll show those too. But this is just looking at normalized intensities, the log2 scale for NECL2, 3, 4, et cetera. And the only thing I really want to point out here is, okay, those 3 replicates, you can see they're pretty consistent. Not perfect, but pretty good. And in some cases, again, like with NECL2 and with MUL4, absolutely nothing was detected in the no turbo control. So, you know, when you get this, you know, this is anything that shows up here is an infinite fold change. And, you know, you can come up with ways to estimate it, but I kind of do agree that in the end, I don't really talk about estimations for most part. I don't find them useful. It's this binary. It's there. It's real. I can believe it. Okay. Okay. We're not going to go through this, but what I will tell you, and it didn't occur to me to do this until like a year after I had, we haven't had all the data for a year, but I think it's really, you know, until I started putting things together in a really organized way, and to get these numbers, you know, to be a week of battling, you know, Excel and doing things, it's just sort of a way of organizing and thinking about things, and then you can start to see patterns and realize, okay, what makes for a good experiment, what doesn't, what are the indicators? So here's the deal. These are our 8 experiments that we've done here. These are the strains that we sent them each time. These are the number of reps. And so what I'm going to talk about is this thing I call percent carboxylase, percent up, percent TurboID only. These are some of the numbers I was indicating earlier. And these are the number of candidates that we got after something I called filtered 1 and filtered 2. So these are the things I want to just briefly talk about. But the main thing is you have to be interested and willing to engage reasonably deeply with the data, not with the raw mass spec data, but at the very least with the Excel spreadsheets that I can get, that you guys send me. Okay, so first is percent up, and that's of course just a volcano plot. We're talking about adding up the red dots, the blue dots, and then dividing red dots over the total. And so percent up is a great indicator of whether your experiment worked. It is TurboID, after all. The whole premise is that everything should be on the right-hand side. But of course, that would be ideal. In reality, it's not going to happen. And even in some crappy experiments where, in some cases, we've had experiments where we couldn't tell the difference between the red and the blue. We still got things that we know were real on the P side. But the more over here, the better. One thing I will say is these are the two proteins that are expressed at the lowest levels, and these never gave as good results with respect to fold change or with volcano plots. So there is some ultimate limit of detection, but if you have a really lowly expressed protein, it's just going to make it harder to detect the signal from the noise. So percent up is useful. So this was percent carboxylase. This is something I actually ran by Dennis and Rick and a few at Sam, I think. You guys took the time to meet with me. So here's the idea, and I haven't heard anyone else talk about this before. So these are those carboxylases that are endogenous that I talked about. They're the ones I was worried would just swamp out everything else. So I would calculate. What I did was I said, gosh, what I did was I said, well, what was the total percentage of carboxylase? I'm going to say the wrong word, reads. You call them IDs. I don't know. Is there a correct word? Abundance. Okay. What is the percentage of carboxylase abundance relative to all the proteins that were detected in that experiment? And you can do that pretty easily just by, converting from log to adding. So you add up these intensities versus all. And the answer is, in a lot of our experiments, somewhere around 20 to 25%. That's sort of the max we ever reach. And in some experiments where I noticed things didn't work very well, in other words, we didn't detect even the bait very well or at the same levels, this number dropped. And what I think happened in those cases is that the beads just weren't washed well. So there was so much just random sticky garbage there that it just diluted out the things that it was supposed to pull down. And so I attribute that to bad samples and bad washing. It's basically always the client's fault. That's what we say every day. I mean, I think it's true. The customer is never right. Okay, so the other thing I will say is, there are labs that have said, gosh, we need to get rid of these carboxylases because we'll increase the sensitivity because they'll waste less time. That's never made sense to me because I feel like, if 75% of the time it's doing the thing I care about, why do I want to go through extra steps? That said, we did go through a bunch of extra steps. It didn't help us at all. Now that may just be us, but I think if people are obsessed with having to get rid of the carboxylases, if you have an easy way to do that, great. But the ways to do it aren't that straightforward, and involves putting tags on all of them and sort of pre-clearing them from the lysate. And then you're introducing more steps and things degrade. I don't think it's worth it. All right, that's my opinion. In fact, I think it's nice to have them there because it tells you something about your prep, about the quality of your prep. The other thing is detected in TurboID only. So in other words, this example here. So how many cases are there when we did these where we never saw it in N2, but we would detect it in our turbos? In theory, you should have some of those, and you should have very few that are detected only in N2, but not in your turbo or only in the non-turbo. And so just like percent up here, this should be a high fraction, this should be close to, a high percent. And what we see is, at least 70% is good, but the range we've gotten is weirdly enough from 10 to 100%. So this one's a little bit more variable. I think this is good enough and that's good enough to be really useful indicators for most people. I'm not going to go into these except to say, I did go to the trouble at least to ask how well these different things I'm talking about correlated. I'm saying, oh, if you find your bait well or prey well, that fold change tells you something about how well you're experimenting. Or if the volcano plot is really asymmetric, that tells you something, right? Or carboxylase. So that's what I'm doing here. I'm actually going to the trouble of showing that there's an R squared. The one funny thing I'll just mention, the only thing I'll mention here that's a little bit surprising, if I plot bait fold change against prey fold change for NECL3 and MOL3, it's not great. You think, if, let's say I did an experiment where the bait was NECL3, finding partners MOL3. Well, if I did an experiment where NECL3 was 50-fold, right, and MOL3 was 20-fold, right? Okay, that's one experiment. Well, what if I did an experiment where NECL3 was 200-fold? You'd expect MOL3 to go up too, because just that experiment works. It's really noisy. The answer is you don't, I mean, I think there probably would be a correlation if you had enough numbers, but there's enough noise in the system that just even the bait and the praise don't always correlate very well. And that's the point I'm trying to make here.
That may be related to your estimating a fold change from not seeing it to seeing it. So that
I didn't do that for these.
oh, okay.
So I didn't use anything where I made up the number.
Okay.
Yeah.
Could it have to do with the amount of each of the proteins in?
It totally could. It could be, so all this variability, is it my worms? Is it the prep of the worms? Is it something to do with the mass spec, or is it all three? Probably it's all three, right?
Well, I just meant it's like the expression of those proteins. Is one of them expressed higher than the other? Not the TurboID, but is there more of it present in one?
Well, yeah, I see. Well, what I'm saying is it sort of shouldn't matter. In terms of the fold change up, it shouldn't matter. Because we don't care about absolute changes, is I guess what I'd say about that. But whether in a different two experiments, there's, you know, it's a puzzle to me, but there's just noise in the system, and that's, you got to deal with it. Okay. Then the question is, how many biological replicants do I need? This is a really tough one. Okay, here's what I know. These are horrible Venn diagrams, but I'll show you something here. So this is experiment 1, NECL3, NECL2, experiment 6, NECL2, NECL3. So the first place you probably want to look is, well, what was specific to NECL3, right? Okay, so, and I should mention, I'm looking at all proteins detected, not just things that are two-fold up. All right, you look here, you're like, okay, there's some overlap. Obviously, there's a lot of overlap in general because each experiment had an overlap of 2,378 proteins. I should have put the total proteins on here. I don't have that. But there is an interesting effect here that I want you to notice. NECL2, NECL3, and the no turbo N2 have 362 in common. These have 516 in common. This experiment shows, a different experiment, shows something similar here between MOLT4 and the no turbo control. In other words, what I think I'm seeing here are somehow their batch effects with biological replicants. For some reason, experiment 1 did a really good job of detecting these 362 proteins, but experiment 6 did a really good job of detecting these 516 proteins. Now, it might have been my preps or something that was up in or present there. I don't know if this could be, is this possibly a mass spec phenomenon?
Could be. It could be a trace sensitivity, because if those proteins you would assume are going to be the most abundant in the cell because they're likely background because they're found in your control and your. So if they're very low level at the detection limit, that could be a instrument maybe a little bit more sensitive one day and the other gets a few more proteins.
What I can tell you is that these proteins here, you don't really care about anyway, because it turns out they're really not the things that end up making your list of upregulated proteins. So even though it's a little bit weird to see this batch effect or whatever you want to call it, it's probably not going to affect your final list. Okay.
So Dave, for this, replicants, is this a pool of worms? Because the worm is very small, right? It's not really a single worm, right? Okay, so you have a
But the worms are
pool.
about the we keep them in alligator pens and move them around with pitchforks. Yeah, they're minuscule. So we're talking thousands, maybe hundreds of thousands of worms that are present.
Okay,
And they're all at different stages.
okay,
And so again, that's casting this wide net. And if there are slight differences in the stages of our populations, which of course there are, there's a little bit more of the little ones and a little bit fewer, that's going to affect to some extent what we get out of it. And I recognize that. Okay, so here's a really kind of the main thing I've been wrestling with is how do I filter out non-specific garbage? That's probably a question you'll get a lot. So first thing, just to keep it very simple, I said filtered or first round of filtering. It has to be detected in all of the bait TurboID replicates, 3 out of 3. It has to be twofold upregulated relative to the no turbo, and has to have an adjusted p-value of less than 0.05. So no reinventing the wheel there. Or it has to be detected in all the bait turbo replicates, but not in any of the no turbo replicates. So this might have ended up being in typical experiments a few hundred. This might have ended up being about 50. But some of these were the absolute best stuff we found.
That's just presence absence.
Yeah. That's twofold. This one is just presence absence, exactly right, but consistent presence absence. Okay, so this sort of thing will eliminate some and/or small or random effects, but it still keeps a lot of players in the game. Okay, so this, however, was not. I got 5 minutes. All right, boy, oh boy. All right, so I'll get through this as well as I can here. All right, so this wasn't good enough is the problem. And so what we did, we used this non-bait specific control, and I did 3 experiments. 2 of them looked pretty good to me, and they had overlap. So of the overlap, 212 had an average fold change of more than 2-fold, and they correlated pretty well too. Okay, those proteins that were in common. So then what I did is I made up my own list of garbage, right? So what I did was I used, again, this non-specific GFP turbo that was expressed in the epidermis. I said, okay, how many of these are upregulated 10-fold versus just the no turbo control? They came from different groups. Sometimes, it wouldn't be detected in one of the N2s, but it always had to be detected in both of the GFP turbo samples. So I made that a criteria. That's why I have these different things. So there's the 10-fold guys, the 5 to 10-fold, the 2-fold, and things that were only detected in the GFP turbo and never detected in N2. That's another 27. So 288 sort of garbagey things to varying degrees. All right. So then I would annotate my spreadsheets. This, I have dozens of these. But I think this is what it takes. So if you look at the top, this is pretty good. This is NECL 2. What's the #1 thing that wasn't detected in the wild type, but was detected always in my turbo? NECL 2 is #1, abundance. One of its binding partners, MOLT4, is #2. And so, this is me just like sort of going from the top to the bottom. And then I annotated everything based on that color scheme I was telling you about, right? And one thing I got to say is, you might think, okay, as we go down, okay, their fold change is going down. That's how it's arranged. But look, there's NECL 3. That's real. That's only like 2 points. And look at this, barely hanging on to the bottom is MOLT3. This does not show up by accident ever. So here's a perfectly good thing that I know is real at the bottom of my list. It tells me I can't throw off things that are below that. And that's pretty much the tail end. So there's good stuff and bad stuff all over the place. And so what I did, I'll just very quickly, I'll just say I plot this so you can see how much potential garbage there is here, right? Either in overlaps or in totals. You'll notice if I do all 3 of these, there's 153 in the overlap. Interestingly, this contains the best stuff and the worst stuff. It's the worst stuff because it's the garbage crap that always happens, and it's the best stuff because this is a complex, so they're all hitting the same thing. Makes total sense, right? And so what I did was
sounds perfectly logical. I mean, it makes sense.
Yeah.
Yeah.
Yeah. So what I did was my filtered round 2, just to eliminate the numbers of things, was to remove everything that was red and orange. And then I calculated the ratios of the fold change with my specific to non-specific, if that makes sense. And basically, I didn't make it too hard. I said it had to be the specific had to be 1.5 fold greater fold change than the non-specific. That allowed me to throw out about half of the yellows. And so I was working with more decent numbers, I'd say. Okay. This, I'm kind of going to go through like ridiculously quickly. The question is, how many biologic replicates do I need? And here's my answer to that is one good one. One good one trumps a half a dozen mediocre ones. And let me show you why I say that. Don't worry about these, okay? Take a look at this. This is just Neckel 2. So we did this experiment 5 times. So how many things showed up 5 out of 5 times? Only 3 proteins. That sounds bad, right? It sounds, oh, your experiment didn't work because it's not reproducible. Well, okay, 2 of those things are part of the complex, and then there's another protein. Okay, how many showed up 4 out of 5? That's pretty good. Well, 1 member of the complex and some other things that might be good, right? And I know they're good. And then there's 3, 5, 2, 5. Well, look at this though. 1 out of 5. One golden experiment got these 2 things. It's this little green sliver that you can't really even see. But those are good. That's a real hit. This did not happen by chance. And you know, you look at other things like this, and it's the same story. Yeah, there's good stuff throughout. It doesn't show up in every one. And one thing I can tell you is that the experiment that worked really well, where it showed up in the one, that was the same experiment for both of these proteins. So the experiment where the gel looked not very promising, brilliant experiment. Couldn't have been better. All right, so I'm going to finish with quick key take-homes. Okay, you can't know how well the experiment worked until you try it. Once you have data, you can determine roughly how well the experiment worked using several parameters. You might not need more than 3 technical replicates. Virological replicates vary more, but 1 good experiment is key. Useful to have a non-specific TurboID control to help filter out things. You need to play with the data and figure out how to filter it. And it might be a case-by-case basis. For us, the C-terminal fusions worked a lot better. Completing carboxylases, no good. You need to have good follow-up studies. And I would argue that AlphaFold is king. I've spent a lot of time with it in the last 1.5 years. I have a lot of opinions about it. I'd be happy to share those someday, but leave it off for now. DIA was key, although you got me thinking about more directed stuff, as you know, possibly as confirmatory. And really, actually, this resource is freaking awesome. Okay, so that is appreciated. These are some of the student, postdoc, and technician who did some of this work. And with that, I'll just thank some people for strains. But other than that, thanks a lot.